Chapter 18, Overview of Motor Systems. This also has the Tracts review at the end as well. So 18 and Tracts. Let's look here. Its lower motor neurons are arranged systematically. Motor control systems involve both hierarchical and parallel connections. So going up, going down, and side to side here. So reflex and, so going up is sensory, but we need sensory in order to have proper motor usually. So reflex and motor program connections provide some of the inputs to the lower motor neurons. Upper motor neurons control lower motor neurons both directly and indirectly. So we're going to look at the association cortex, cerebellum, basal nuclei. They modulate motor cortex. Corticospinal tract. So descending from the cortex to the spinal cord, and then synapsing for our alpha motor neurons out to create movement in our muscles. So corticospinal tract has multiple origins and terminations. Corticospinal axons arise in multiple cortical areas. Think about homunculus man. It has a sensory and a motor component. Corticospinal input is essential for only some movement. Upper motor neuron damage causes a distinctive syndrome, and there are upper motor neurons for cranial nerve motor nuclei as well. So understanding the Brodmann cortical areas here, so you can see that kind of magenta-y pink kind of color. So 4, 6, and 8, that motor functions, 4, 6, and 8. So that's the precentral area to that central sulcus, remember. Okay, so post is going to be sensory, pre would be. This is an excerpt from the book or booklet that I put on your Brightspace resource for Brodmann's areas. So this is area 4, the primary motor cortex. Go ahead and take a look at that and the other Brodmann's areas when you get a minute. I find them very interesting. And remember the exercise we did where if they can't do one of these functions, as I call it, you go in the side door, and whatever function they can do, you strengthen that in hopes of creating neuroplasticity changes for the better. Area 6, that premotor cortex here, you can read up on that. Here's area 8, also known as frontal eye field. Sometimes you'll just see it as FEF, frontal eye. This is a nice slide for you to kind of see all the different connections and how things are interrelated. So association cortex and how the cerebellum and the basal nucleus get involved all the way down to the pyramidal tract on the cord there. So kind of a nice little way to pull it together. So here we have our planning, programming, pathway, tract, voluntary movement, sensory motor functions. I had put in just the definitions here, just in case you needed a little refresher on what those definitions are. I'm not going to read them to you because we kind of know them right now. And if you don't, they're right here for you. Feel free to pause anywhere along the line. The idea behind this is that it's a rapid review. You can kind of check yourself here. Upper motor neurons and lower motor neurons. So upper motor neurons are located in your brain and spinal cord. They send signals to lower motor neurons. Lower motor neurons are in your brain stem and spinal cord as well. When they get a signal from the upper motor neuron, they send another signal to make your muscles contract. And you can see the picture on the right. So difference between upper motor neuron lesions and lower motor neuron lesions. So imagine the patella strike for the reflex. So if we strike the tendon at the knee and we don't get a response, first we think user error, right? We keep hitting the poor patient or our poor partner in lab. And then we go, okay, well, maybe that's a lower motor neuron lesion. So we would see no deep tendon reflex, decreased or absent. And then in an upper motor neuron lesion, we would see an increase or a brisk or clonus. So you might see a really big flare of the knee kicking out, the leg kicking out, or it might just continue to kind of beat to that. That's an upper motor neuron lesion. So this chart goes through the differences, but that's the one that I think of as the easiest one to remember. So upper motor neuron lesions, normal till disuse atrophy. Lower motor neurons, there's going to be atrophy there. Upper motors, not so much because you're still able to use the muscle. The tone is going to be increased though. And then lower motor neurons is hypotonic because it's not activating appropriately. Power reduced in upper motor neuron lesions because you don't have full control over it. And then lower motor neuron lesions are going to be severely decreased. Abnormal movements. Upper motor neuron lesions are going to have fasciculations, are not going to be seen here, but lower motor neurons are going to have fasciculations. So they're due to the degenerative process in the anterior horn cell. So fasciculations can be seen as twitches. They could be normal, they could be lower motor neuron lesions, or they could also be a sign of ALS. The difference here, sometimes people get fasciculations and fibrillations confused. Fibrillations generally not normal. Usually, well, they're really only going to be seen on an EMG, and it's showing signs of atrophy. So the difference that I remember is that I need a machine to see the fibrillations versus fasciculations. You can just see them as twitches. So fibs, okay, fibrillations, fib is a lie, and then they have a lie detector machine. Okay, so the EMG is a machine. So that's how I keep those two words straight in my head. So for fibs, I need a lie detector machine to be able to spot it, but that's the EMG in my little memory aid there. Okay. What's next here? So reflexes, we went through that, and then Babinski is when you scrape the bottom of the foot and the big toe is upgoing or kind of extends out. And then the lower motor neuron lesion, it's not necessarily going to be downgoing. You just might get normal is no reaction, and normal can also be downgoing as well. And the analogy that I used in class, remember, is if you were going to come at me and punch me, which would be really weird, but if you were to come at me and punch me and I extended open into that punch, that's not neurologically normal. That's an upper motor neuron lesion. Babinski is the analogy there of the foot opening up into extension when it's being disturbed. So the normal thing would be to kind of go into a flexion protection response. Each lower motor neuron innervates a group of muscle fibers forming a motor unit. Okay, so lower motor neurons, the target of the central nervous system pathways, and connections involved in motor control are arranged in the spinal cord and brainstem in groups corresponding to individual muscles. Just as there's synaptic maps in sensory pathways and in cortical areas, there's too a systemic arrangement of clusters of motor neurons. Lower motor neurons are arranged systematically in the anterior horn of the spinal cord. So we looked at this when we looked at Rex head lamina as well. Motor neurons and axial muscles are located medial to those for more distal muscles, and those for flexors are posterior to those for extensors. So flexors posterior, okay? Extensors anterior. This axial distal mapping corresponds to the arrangement of descending pathways, some of which are important for postural adjustments of axial muscles and others for the control of more distal musculature. There's a nice picture here. You can see in the gray matter there, flexors, extensors, elbow, wrist, hand, somatotopy of where these things are. Another nice picture here to kind of pull it together. This one I highlighted what I really wanted you to make sure that you saw. The lateral extension here of the anterior horn is where finger motor neurons live, and they, questionable ways on how they are figuring this stuff out. It's research. So we can see the highlighted area corresponding to their research here. So motor control systems involve both hierarchical and parallel connections. The inputs that determine the level of activity of lower motor neurons can be divided very broadly into three overlapping classes. So we have 1, built-in patterns of neuronal connections, 2, descending pathways that modulate the activity of motor neurons, 3, higher centers that influence the activity of descending pathways. So this picture here is over on the right. You can see the association cortex in the middle of that picture. Off to the left, basal ganglia, basal nucleus. Off to the right, cerebellum. So basal ganglia, basal nucleus controls voluntary movements, procedural learning, habit learning, eye movement, cognition, and emotion. So this is the ready to go state. Okay, so we have this constant ready to go, ready to go, ready to go. Basal nucleus is the calming hand on the shoulder saying, hey, not yet, don't go yet. So it controls our movement by inhibiting it. So that picture up on the left there, I think of it like the leash on the dog. Okay, so the person and the leash holding back that dog is the basal nucleus, saying, hey, don't go yet. Our bodies are that dog, always ready to go. Basal nucleus calms it down. So damage to either upper or lower motor neurons or muscles causes weakness and a distinctive set of accompanying symptoms and signs. Damage to higher centers also causes distinctive movement abnormalities. So we're seeing involuntary movements and coordination, difficulty initiating movement, but not necessarily accompanied by substantial weakness. So the reflexes, reflex and motor program connections provide some inputs to lower motor neurons. You might remember this slide. It's kind of weird that he's using a hammer. Please don't use a hammer. Use your reflex hammer. People think we're weird enough, right? The stretch reflex is an obvious and simple example of a built-in pattern of neuronal connections that controls to some extent the activity of motor neurons. Stretching a muscle stimulates its muscle spindles, whose afferent fibers end on motor neurons, which in turn cause the muscle to contract. So those 1A afferents, remember, muscle spindles, 1A afferents, coming into the cord saying, hey, I've been overstretched. Flexor reflex, here's Mr. Sparkle Pants. Remember that slide? The reflex involves several segments and all connections are polysynaptic. In the example shown, a nociceptive fiber, so pain, from the foot enters the spinal cord at S1 and activates through at least one interneuron the motor neurons to iliopsoas and hamstring muscles. Iliopsoas, remember, is going to be your major hip flexor, and hamstring is going to flex the knee. So boom, it's going to flex the hip, flex the knee, you're jumping off of that tack. Crossed extension, so we're going to flex that one side, but we have to jump back onto the other leg in order to really get off of that tack safely and appropriately. So stepping on the tack initiates the flexor reflex in the previous slide, but also excitation through an interneuron of the contralateral antagonistic muscles. In this case, contraction of the contralateral quads helps the leg not puncture the foot and support the body. We use agonist-antagonistic understanding of this system all the time in neuro rehab. So our cross crawls are understanding this. If we have trouble with one muscle, we activate its antagonist. We do this all the time. We use a stretching called proprioceptive neuromuscular facilitation, or PNF stretching uses this, neuromuscular reeducation, all sorts of fancy words for it. But it's really just an understanding of lighting up that antagonistic muscle in order to alleviate the agonist. Finally, there are networks of interneurons in the brainstem and spinal cord that can act as pattern generators for rhythmic movements such as breathing and walking. Although some of the same interneurons involved in reflexes may also be part of the circuitry of these motor programs or central pattern generators, or what you'll see called CPGs. These programs are more than simply a stringing together of reflexes, each one triggering the next. One indication of this is the observation that the principal features of CPGs can persist in the absence of afferent input. And so this kind of program, pre-programmed stress response that kind of sticks around. Principal locations and projections of upper motor neurons. Only one corticospinal fiber is indicated and is shown crossing the midline to join the lateral corticospinal tract. However, some corticospinal fibers, mostly headed for lower motor neurons for axial muscles, do not cross in the pyramidal decussation and join the anterior corticospinal tract. And then those abbreviations are for the pictures in the book. Okay. Association cortex, cerebellum, basal nuclei modulate our motor cortex. So even though corticospinal, rubrospinal, remember flexion protection, reticulospinal, some basic stuff in there, vestibulospinal, balance, are able to influence lower motor neurons and their local connections, this still does not explain how a voluntary movement is made. There's activation of multiple areas of the frontal cortex, which occurs up to 2 seconds before a voluntary movement is made. So a planning stage, okay? And that All such movement has an afferent component, so sensory is coming into that area. So even though we try and separate these things out based on the central sulcus, so pre-central area is motor, post is sensory. Is anything really that cut and dry and narrow? No, it's never just black and white, right? So we have to have a sensory input to understand how to move appropriately. Okay, so we need a little bit of a two-way street happening there. We can specify some of the structures and connections that must be involved because damage to these structures and connections results in defective movements. Cortical spinal tract has multiple origins and terminations. During the 19th century, it was discovered that electrical stim, E-stim, of certain areas of the brain causes movements of the contralateral side of the body. So we stim one side of the brain, we get the other side of the body to move. That's how we figured out that it crosses over, it decussates. In humans, the area with the lowest threshold for this effect lies in and near the anterior wall of the central sulcus. So this area has been, we call this area the primary motor cortex. Primary motor cortex. Here's good old homunculus man. So again, medial aspect of that is lower extremity, then upper extremity, then face and tongue, further out lateral. And you can see where that is on the brain in the colors there, green and purple. Another picture here reminding us of the central sulcus and the pre and the post central areas. Again, remember, not all cut and dry, but you answer it as though it is on your boards, okay? This here is just giving the connections for the brain. You can check that out if that is helpful to you. Basically just what we're saying in a picture instead. Connections of the cortex with the basal ganglia, basal nucleus, and cerebellum, programming of complex movement. So the pyramidal motor system, primary motor cortex and the pyramidal tracts. Okay, so pyramidal tracts is assisted by the basal nucleus and cerebellum in the planning and programming of complex movements. Again, we need a planning stage, and we're getting sensory input in from a planning stage, while afferent fibers of the motor nuclei project directly to the basal nucleus without synapsing. The cerebellum is indirectly controlled via pontine nuclei. The motor thalamus provides a feedback loop for both structures. So the efferent fibers of the basal nuclei and the cerebellum are distributed to lower structures, including the spinal cord. The importance of the basal nucleus and cerebellum in voluntary movements can be appreciated by noting the effects of the lesions in these structures. So while diseases of the basal nucleus impair initiation and execution of movements, think Parkinsonism, cerebellar lesions are characterized by uncoordinated movements, so reeling movements of inebriation. Okay, so think of somebody that you've seen that had too much to drink and they're kind of rolling around, caused by a temporary toxic insult to the cerebellum. And you could see this schematic here. So our volitional movement requires constant feedback from our muscle spindles, tendon organs. This is how we know where we are in space. So the stretch receptors basically of our body saying, hey, this is where you are, and stay within, as it's saying, your desired limits. Don't overstretch. So many, this is so important to understand as a chiropractor. Our injuries are usually caused by an overstretch to this system here. So we overreach, the spindles send a signal saying, hey, you've overstretched, lock it down. Okay, so the cord is going to tell it to then lock it down. And then it's not going to move appropriately. So let's say you stretch your arm out a little too far, and then those spindles are like, oh, I'm overstretched here, I'm afraid I'm going to tear. So the cord's like, okay, lock it down. It doesn't lock down in a good alignment. No, it's twisted slightly, and then it's locked down in that rotation or that tilt, that lateral tilt. So we come by, see that it's not moving, that it's a body right or maybe a PLI, depending on what listings you like to use. Then you're like, all right, well, I'm going to come in here and I'm going to quickly adjust this, and you need to be quicker than the signal from that muscle spindle and that GTO. That's what we're trying to do. So we can spindle down those muscle tissues is what it's called when we're therapeutically, instead of stretching it, because it's been overstretched and it's creating a tension, saying, hey, I'm overstretched, lock it down for safety's sake, I don't want to tear. So if we go in there and stretch it some more, that it feels good because the stretching stops the pain signal, but it's actually going to be the opposite of what we need. So you spindle down or push the muscle spindles together ever so slightly, and then that sends a signal of, oh, hey, I'm not overstretching anymore, I can unlock, and then you adjust it. There you go. That's what we do. That's the whole idea. Okay, so repeat from the last slide that voluntary movements require constant feedback from the periphery in order to remain within the desired limits, and I explained what that means to us in the previous slide. Because the motor and sensory systems are so closely interrelated functionally, they're often described jointly as sensory motor, okay, sensory motor system. The spinal cord, brainstem, cerebellum, and cerebral cortex are the three control levels of the sensory motor system. All information from periphery, cerebellum, and the basal ganglia passes through the thalamus on its way to the cerebral cortex. The clinical importance of the sensory system in movement is illustrated by the sensory ataxia that may occur when sensory function is lost. The ocular motor component of sensory motor not shown in that schematic. So what are we looking at here? Basal nucleus, more inhibition than excitation. It inhibits the thalamus. Reciprocal inhibition in the cord, very important for movement. Altered locomotion and gait. Spinal cord dysfunction, altered locomotion due to feedback inhibition. Basal nucleus, so globus pallidus and putamen inhibits the thalamus. Thalamus is excitatory to the cortex. Cortex is excitatory to the corpus striatum. Corpus striatum is the caudate nucleus and the putamen. Okay, so corpus striatum is the caudate and putamen. Movement must have disinhibition. So remember, it needs that leash on the dog in order for it to have meaning. Globus pallidus and putamen calms the thalamus. Excitatory is acetylcholine, nicotinic acetylcholine receptors, acetylcholine, musculoskeletal. So GABA, dopamine, and serotonin are going to be inhibitory. And then Purkinje cells in the cerebellum use GABA to inhibit. So this is just like kind of a random slide I threw in there of like things I saw as test questions that I wanted you to associate, word association. Okay. Corticospinal axons arise in multiple cortical areas. So you can see that diffuser tensor image over there of corticospinal. Corticospinal tract conducts impulses from the brain to the cord. It contains mostly axons originating from the motor cortex. Corticospinal tract is made up of two separate tracts in the spinal cord, so lateral and anterior corticospinal tract. Corticospinal tract also contains Betz cells, B-E-T-Z, Betz cells, the largest pyramidal cells. That's a test question I've seen before as well. So largest pyramidal cells of Betz, B-E-T-Z, which are not found in any other region of the body. An understanding of these tracts leads to an understanding of why one side of the body is controlled by the opposite side of the brain. Giant cells of Betz. Betz cells are concentrated specifically in the primary motor cortex. Primary motor cortex area 4. Okay? But only about half of the corticospinal fibers originate in this cortical area. The remainder come from the adjacent frontal motor areas and from the parietal lobe, particularly somatosensory cortex of the post-central gyrus. Remember sensory with that S for post-central? In view of this fact, the cortical complex on both sides of the central sulcus is sometimes referred to as the sensory motor cortex. I like this slide. I'm a sucker for anything that's moving like that. Gives us a nice perspective. It's that diffuser tensor imaging again. Main key point here for test questions is pyramidal tracts are both corticospinal and corticobulbar. Okay, so corticobulbar, corticospinal, also known as the pyramidal tracts because of those pyramidal cells of Betz. Corticospinal tract, discrete, voluntary skilled movements such as precise movement of the fingers and toes. This is imperative in understanding that the left hemisphere of the brain controls the right side of the body, while right hemisphere, vice versa, of the brain controls left side of the body. So cortex, opposite side, cerebellum, same side. So right side of the body, right cerebellum, left cortex, and vice versa. The tracts cross in the medulla oblongata, so at the decussation of the pyramids of the medulla oblongata. The cortical bulbar tract carries information to motor neurons of the cranial nerve nuclei rather than the spinal cord. Cortical bulbar, cranial nerves. Cranial nerves are ipsilateral, except for cranial nerve 2 is ipsilateral and contralateral is kind of a special case, remember, rather than the spinal cord. Okay, so these nerves control the muscles of the face and the neck, are involved in facial expression, mastication, chewing, okay, trigeminal chewing, swallowing, and other functions. Here's a little side-by-side for you to study the pyramidals versus the extrapyramidals. And then this was a project that we did, kind of putting it all together here. So motor pathway, corticospinal tract originates from pyramidal cells in layer 5 of the cerebral cortex. About 31% of its fibers arise from the primary motor cortex. Other contributions come from the supplementary motor area and the premotor cortex, which constitutes about 29% of the fibers, while the somatosensory cortex, parietal lobe, and cingulate gyrus of that limbic lobe supplies the rest. The average fiber diameter is in the region of about 10 microns. Around 3% of fibers are extra large, 20 microns, those pyramidal cells of Betz, mostly in the leg area of the primary motor cortex. So remember homunculus man, leg area, medial. Neuronal cell bodies of the motor cortex, together with their axons that travel down through the brainstem and spinal cord, are commonly referred to as upper motor neurons. So it crosses over and then synapses. So our premotor cortex here, primary motor cortex, is neither the only source of the corticospinal fibers nor the only cortical area involved in motor control. There are also maps of the body in the premotor cortex directly anterior to primary motor cortex and in the supplementary motor area, located on the medial surface of the hemisphere just anterior to the representation of the foot in the primary motor cortex, so medial area. Nice picture here for you to kind of go through. You can pause and go through that. Cortical spinal axons rise in multiple cortical areas. We're good with that, so go back to those areas if you need it. Again, kind of speeding this up because it's supposed to be a review. Upper motor neurons control lower motor neurons both directly and indirectly. You can look back at that if you need to. Upper motor neurons for cranial nerve motor nuclei. So some corticospinal fibers end directly on spinal motor neurons. Some end in the posterior of the dorsal horn and the intermediate gray matter or interneurons of the anterior horn. Those not ending directly on motor neurons have a variety of effects, ranging from regulating the access of info to ascending pathways to affecting the activity of motor neurons via interneurons. Other fibers leave the cerebral cortex, descend through the internal capsule immediately anterior to the corticospinal tract. Cortical bulbar, commonly used to refer selectively to those fibers that affect the motor neurons of cranial nerves. Most, however, act on interneurons of the reticular formation. The oculomotor, trochlear, and abducens nuclei receive no direct cortical bulbar fibers. There are other peculiarities about the innervation of these nuclei, so they're in a different chapter. So the following discussion pertains to trigeminal, facial, and hypoglossal motor nuclei. So our nucleus ambiguous, remember ANT, ambiguous, 9, 10, ANT, and spinal accessory, so spinal accessory, cranial nerve 11, SCM, and Tracts. Cortical bulbar. These nuclei receive bilateral cortical bulbar innervation. Fibers originate from the face and mouth of motor cortex and other areas of the frontal and parietal lobes to the bulbus, so more modernly known as medulla oblongata. Bulb, medulla oblongata. Cortico-bulbar is cortex to medulla oblongata. Responsible for regulating brainstem activity. Influences the overall activity of the pons and medulla oblongata and activity of the motor component of cranial nerve nuclei. So cranial nerve 3, 4, motor division of 5, and then 6, 7, 9, 10, 11, and 12. They accompany corticospinal tract almost to the level of the nucleus they influence, where they part company with corticospinal and end at the appropriate motor nucleus on both sides or in the adjacent reticular formation. Hence, there's no single cortical bulbar decussation, analogous to the pyramidal decussation of corticospinal. Major exception to this general pattern involves facial motor nucleus, as we looked at in chapter 12. So motor neurons of the lower facial muscles are innervated mainly by contralateral cortex, corresponding to the way we can contract muscles around the mouth asymmetrically. Upper facial muscles are innervated bilaterally. The result is that an individual with a unilateral cortical bulbar medulla, damage, a lesion in one cerebral peduncle, for example, is unable to smile or show their teeth symmetrically or puff out their contralateral cheek. However, the ability to blink and wrinkle the forehead on both sides remains. Hence the stroke aspect of stroke is eyebrow sparing. Okay, so this would be. This is the slide that's showing the tongue. What an effect would have from a stroke on the tongue here. So tongue weakness caused by damage to cortical bulbar fibers in the internal capsule. So the 68-year-old woman with high blood pressure referred to neuro after experiencing sudden weakness and chewing and mild dysarthria. So remember when we played Selma Blair and she had trouble speaking, that kind of rigid, kind of ratchety speech, that's that dysarthria. When she was asked to protrude her tongue, a left lingual paresis was evident. Weakness of the left side of the tongue resulting in deviation of the tongue to the left is usually the result of damage to the left hypoglossal nerve or nucleus. In this case, the only abnormality revealed by CT was a small hemorrhage in the posterior limb of the right internal capsule. Okay, so you can see it in the right there if you note that blue arrow pointing to that white mark. Hypoglossal and trigeminal motor nuclei and those neurons of the spinal accessory nucleus that innervate trapezius receive some input from the cortex of both hemispheres. Input is mostly contralateral, so it crosses over. Hence, after damage to the motor cortex, internal capsular cerebral peduncle, also known as crus cerebri, on one side, there may be slight and typically transient weakness of the contralateral trapezius, the upper trap area, and masseter, one of the chewing muscles, and the contralateral side of the tongue. I put this in here because it's good to see how we test it and what the deficits might be. So posture and abnormal movement. So you would ask your patient to hold the arms outstretched with your eyes closed. We call it a pyramidal drift. Okay? If the tendency for the hand to move up, and supinate if the hands were held outstretched in a pronated position, or to pronate downward if they started in a supination. So if you start in one direction and it moves into the other direction, pronated or supinated, we call it a pyramidal drift. Pyramidal tracts, again, being corticobulbar and corticospinal. We call it a cerebellar drift, generally upward and with excessive rebound movement. So remember when we looked at the test where they were going to punch themselves in the face, don't punch them in the face though. Modify it the way that we showed you. Rebound movements if the hand is suddenly displaced downward towards the examiner. Parietal drift is an outward movement on displacing the ulnar border of the supinated hand, so outward movement. These are some really interesting books if you get into this aspect of neuro rehab, which I highly suggest you kind of check out. Vestibular rehab and balance and dizziness. Your patients will thank you for being able to fix that, or at least help with that, hopefully, right? All right, so here's our boards. Okay, so boards review slides. So 6 ascending tracts and 6 descending tracts. So the way we look at this, I worked this out on the whiteboard when I was going through my NeuroDip program and also looking at what the boards are expecting of you. So this is the high yield test question stuff that I kind of came up with meshing those two things. Okay, so from my teacher, who is Dr. Dishman, who does the NeuroDip program. So credit where credit is due there. Okay, so 6 ascending, 6 descending tracts. So the ascending tracts, so if you look at this, I'm going to underline it for you. So the way I did this was I just wrote it out on the whiteboard over and over and over again until I had it memorized. Okay, so ST, DC, and SC. So I would take the S here and the C here, and that's my third one here. So that was first and foremost this. So this is spinal thalamic. The ALS is also known as spinal thalamics. And then the DC being dorsal columns, and then the spinocerebellars. So we first have to know what we're describing. So we're going to describe the ALS, or the anterolateral system, spinal thalamics, dorsal columns, and spinocerebellars. ALS has lateral and ventral. Dorsal columns has fasciculus cuneatus and fasciculus gracilis. Spinocerebellars have dorsal spinocerebellar and ventral spinocerebellar. Then we're going to memorize 6 of our descending tracts here. So they start with the letters C, V, R, T. So cortical spinal has 2, vestibulospinal has 2, and then there's rubrospinal and tectospinal that we need to know. So if you note, right here, we have lateral and ventral in the ALS. We have lateral and ventral across from it here in our corticospinals. And then lateral and medial for vestibulospinal. So let's get into it, shall we? ALS, or our spinal thalamics, so we have a lateral and we have a ventral. So the way to read these slides is when we separate them out, so lateral is pain and temperature, ventral is light, touch, and pressure. Both we just memorize as touch, pain, and temperature. Okay. And then again, both at the bottom there, at the middle of the slide really, we want to understand that they cross immediately. Where are they crossing? So it's coming in, synapsing at Rexhead lamina 2, or that substantia gelatinosa, that nociceptor comes in from the receptor level at the skin, right? Comes in, synapses at substantia gelatinosa or Rexhead 2, and then it's going to cross over, and then we're calling it spinal thalamic tract. It's going all the way up to the VPL of the thalamus. So the nuclei of the thalamus called the VPL. And then it's going up to its appropriate area in the somatosensory cortex here of Brodmann's 312. So think about homunculus man on where it's going to go. So spinal thalamics, touch, pain, temperature, crosses immediately at substantia gelatinosa, because substantia gelatinosa we memorize as touch, pain, temperature as well. Goes to VPL of the thalamus, so does dorsal columns, and then up to its appropriate area in homunculus in Brodmann's 312. Same as dorsal columns. Here's our dorsal column. So fasciculus gracilis, think of the muscle gracilis, that's lower extremity. Fasciculus cuneatus, upper extremity. Both are going to be for conscious proprioception, kinesthesia, vibration, and 2-point discrimination. Ascends on the same side in the cord, crosses at the internal arcuate of the medulla, ascends via the medial lemniscus, so does spinal thalamic, but most of the time we associate it with dorsal columns, the DCML. And then synapses there at the VPL of the thalamus and up into Brodmann's 312, and think about homunculus man and where exactly it's going. So that bottom half there, those last 3 components, same thing as spinal thalamic. Spinocerebellar, okay, spinocerebellar, unconscious proprioception, muscle spindles and GTOs, 1As and 1Bs, remember. So dorsal spinocerebellar versus ventral. So dorsal is ipsilateral. First order neuron is in Rexhead 7 or Clark's column, and it's going to go up and into the inferior cerebellar peduncle over to the cerebellum that way. Ventral spinocerebellar is contralateral. No Clark's column, we just call it the dorsal horn instead. And then it's going up and in through the superior cerebellar peduncle. So the differences here, ipsilateral versus contralateral, Clark's column versus dorsal horn, inferior cerebellar peduncle versus superior cerebellar peduncle. Here's a picture of that homunculus on the right, and then on the left in green is that 312 area. So right at that separation there between the yellow and the green to the right, that's that 312 or the post-central area. There's another picture here for you, so you can hang out with that for as long as you like and pause the recording. Corticospinal tracts, so corticospinal descending. Okay, so descending, corticospinal tracts. So both our lateral and ventral are going to start off primarily at our BET cells, our pyramidal big cells in the primary motor cortex. we're looking at Brodmann's 4 and 6. Goes down through the corona radiata, through the posterior limb of the internal capsule, through the cerebral peduncle, also known as crus cerebri, down through the anterior pons, crosses over at the pyramids of the medulla, at that decussation of the pyramids of the medulla, and then synapses on alpha motor neuron in the gray matter, or what we call Rexed 9, remember. And then separately, so lateral is going to be crossed, and that's fine motor. The ventral, or 10 to 20% of the fibers here, are ipsilateral, same side, and it's gross motor. Here's a little reminder of the BET cells and a cool picture of it there on the right for you, and the different layers are even pointed out. Vestibulospinal tract, so there's a lateral and a medial. If you note at the bottom there, board's info, the same for lateral and medial. So we're just going to memorize that it's ipsilateral, or same side, through the medulla. Synapse is at the motor neurons in the anterior horn. And this is coordinating our head, eyes, posture, and balance. So as we're moving, walking, standing, looking around, our eyes and our balance are being coordinated this way so we don't fall over when we turn our heads, right? Tectospinal, tectum. Okay, so what's the tectum, or what areas are on the tectum? So superior and inferior colliculus are part of the tectum area. So this one here is superior colliculus of the midbrain, crosses over right there at that superior colliculus, descends medially through the pons and medulla, terminates on the motor neurons of the cervical cord only, so neck, okay, because it's going to turn the head to turn and look, remember, as part of that reflex. So superior colliculus, you see something out of the corner of your eye, you turn and look. That's why we're going to turn and look and have this only synapsing at the cervical cord. Head and neck movement to that visual reflex. Rubrospinal is going to be the flexion protection aspect that can be coordinated with that as well. Rubro, ruby red. Okay, so that's the red nucleus. So the trickery here is you'll see it called actually sometimes the dentorubrospinal tract, because it's actually starting off in the dentate nucleus, but they just didn't really, the renaming of it didn't catch on so much, I guess. So it starts off in the cerebellum at the dentate nucleus, and then the red nucleus in the tegmentum of the midbrain, and then decussates immediately through the ventral tegmental decussation. Remember, tectum in the back, tegmental area is more anterior. Descends through the pons and the medulla, synapses on the alpha motor neurons, so Rexed 9 in the anterior horn of the cervical region, and this function is fine motor control and muscle movement. And when this area undergoes a stroke, when this is affected, that's part of what we call a Benedict's stroke, an unwanted movement of the upper limb. So if corticospinal is going to be affected, we're going to see a paralysis of the upper limb, lower limb as well. But in if rubrospinal is affected, so if it's a Benedict stroke, we're going to have unwanted, uncoordinated movements of the upper limb instead of paralysis. That's the differentiation between that Weber's stroke, okay, which is the paralysis, and then Benedict stroke, which is the unwanted movement of the upper limb. Both are going to affect cranial nerve 3 as well, though. Okay, that is the conclusion of Chapter 18 plus Tracts. So that last little bit there, I'd highly recommend checking that out before taking your boards part 1. Hopefully it'll help you out with everything that you need to know on Tracts, and I will see you for the next chapter.